Hello dear students, I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear students, we are in continuation of the chapter that is microbes in human welfare. In previous class, we have already talked about the microbes in household products. In today's lecture, we are going to talk about microbes in industrial products. And when we talk about the microbes in industrial products, we know that in industry, microbes are used to synthesize a number of products which are valuable to human being like beverages and antibiotics. Okay, so in industrial products, even in industry, microbes are used to synthesize a number of products valuable to human being, and it include mainly beverages and antibiotics. So you can see this is the internal industrial production of some of the important components for the human health and human being. So if we talk about examples, your examples are the beverages and antibiotic. Now, generation of these useful products on an industrial scale require a growing microbe in a very large scale, or we can say in a very large scale in large vessels, and these are called fermenters. So what the production of this component like beverages and antibiotic is carried out at industrial scale in a large vessels, very large vessels, which are called fermenters. And this fermenter somewhat look like this, in which these microbes are used for the proportion of, for the preparation of these products. So if we talk about first fermented beverages, dear student, microbe, especially yeast, have been used for the production of beverages like wine, whiskey, brandy, and rum. Okay, for the production of these alcoholic beverages, we can say yeast, which are the unicellular fungus, mainly used for the production of this. And if we talk about this yeast, which are going to produce this beverages, in this, if we talk about the yeast, which we call baker yeast, also that is Saccharomyces cerevisiae, which is used in baking industry for the bread making, as well as it is also used for the alcohol production. So it is also known as brewer yeast, and it is also known as brewer yeast or beer yeast, and it is used for the fermenting malted cereals, okay, and fruit juices to produce ethanol. Okay, so for alcohol production, this yeast is used. So it is known as baker's yeast, brewer's yeast, or beer yeast. So this is the brewer yeast, which is available in the market in the form of powder, okay, which can be used here. And the ethanol is produced, alcohol is produced. So depending on the type of raw material used for the fermentation and the type of processing, that is with distillation or without distillation, different type of alcoholic drinks or beverages are obtained. Okay. Now, if you want to use this different raw material, and if you want to you produce with distillation and without distillation, so through the distillation, the concentration of alcohol is getting increased. So whatever the alcohol beverages which are produced without distillation will have a less concentration, that is less than 13%. But those beverages which has a more than 13% and high alcoholic concentration, they are produced through the distillation. So wine and beer produced without distillation, so they have a concentration less than 13%. Okay. But other like whiskey, brandy, rum, gin, vodka, they all have a concentration higher than 13% because they are produced through the distillation. So whiskey, brandy, rum are produced by the distillation of the fermented broth. Okay. So they have a high alcohol concentration. So before discussing antibiotic, we should talk about some more things about this beverages. So if we talk about these products in detail, then we have to note down their concentration and all these things. Okay. So let's discuss about that. So first we remember that the Saccharomyces cerevisiae we have studied, that is a brewer yeast or beer yeast, but here some more species are there that you should remember. Saccharomyces ellipsoidans. Saccharomyces ellipsoidans, if we talk, These are wine yeast. These are wine yeast. And after this, Saccharomyces sec if we talk, they are sec yeast. And then Saccharomyces pyriformis is known as ginger or yellow yeast. Okay. Now the distilled and undistilled products are there. The naturally fermented alcoholic beverages have alcoholic concentration less but high in the distilled products. So if we talk about the alcohol, the substrate which is used for them and their concentration, then we can differentiate them properly. So if we talk about the type of alcohol beverages, then substrate used and concentration of alcohol, then it is known that the alcohol beverages, first we are going to talk about beer. Now beer, for the production of beer, the substrate which is used, that is barley malt, and it has 3 to 6% alcoholic concentration. Second alcoholic product is wine, and here it is obtained based on the substrate like fruit juices, and it has 9 to 12% alcoholic concentration. And these are undistilled beverages. The third type of alcohol we are going to discuss, that is whiskey. I hope you heard the names. This is produced based on the substrate, that is fermented cereals. And here it has 50% of alcoholic concentration. Then fourth is brandy. And brandy, for brandy substrate used, that is the fruit juices, and it has 60 to 70% alcohol concentration. Then the fifth beverage is the rum, and the substrate used are the molasses, and here the alcoholic concentration is 40%. Then if we talk about gin, that is based on the rye malt substrate, and it has 40% alcoholic concentration. But if we talk about vodka, in the potato is used as a substrate, and it has 60 to 80% alcoholic concentration. Okay, so such a way these fermented beverages are produced. Now, apart from fermented beverages, the antibiotic is the another thing which is produced at the industrial level. So, if we talk about antibiotics, what are they? First, the question is, so antibiotics produced by microbes are regarded as one of the most significant discoveries of the 20th century. Okay, it is considered the most significant discovery because what are these antibiotics? So it is known that antibiotic, if we talk, it is obtained from the Greek word, and here it means against, and bio means life, means together they mean against life. Okay, so the antibiotic refers to the particularly the thing which is used against life. So now anyway, these antibiotics are chemical substances which are produced by some microbes and kill or retard the growth of other microbes, or we can say disease causing microbes. So they are produced by a microbes and against the life, that is against microbes, so the disease can be reduced. Now, so they are chemical substances which are produced by some microbe and kill the retard the growth of other microbe, that is the disease causing microbe. Now we are familiar with the commonly used antibiotic, that is the penicillin. We know about that. Okay. Now we remember If we talk about the suppose we are taking an antibiotic and killing a one microbe which is infectious to our human body. So for that microbe which is infecting or causing disease, for this the antibiotic is acting against life. But if we talk with respect to human being, then for us this is pro life. This is not against life. Okay, this antibiotic which is beneficial to us, this is a pro life with respect to the human. Now we are going to talk about the penicillin, which is one of the known antibiotic. And the first and foremost question is that we can ask, that is, who discovered penicillin? So we know that the penicillin is the first antibiotic which is discovered by Alexander Fleming. Now the question is how he discovered. So penicillin was the first antibiotic to be discovered, and Alexander Fleming discovered is, now he discovered this penicillin as the first antibiotic when he was working on a staphylococci bacteria. Okay, and once he observed that, suppose what is the theory history behind this? The Alexander Fleming was working on this staphylococcal bacteria, and he made several culture plates by using this bacteria. And some of them used plates they left in the basin, and then he gone for some work. After few days, he come back and see this particular petri dish. Then he see that one dark mold or dark structure is going in the culture plate. So he observed a mold growing in one of the culture plate, and around which he observed that Staphylococcus bacteria could not grow. So he observed a mold growing in one of the unwashed culture plate around which Staphylococcus bacteria is not able to grow. So later on he found out that it was due to the chemical which is produced by the mold or the fungi. And later on he named it a fungus, that is a penicillin, and he named this antibiotic as a penicillin, and it is obtained from the mold or a fungus, that is Penicillium notatum. Okay, so first antibiotic, first penicillin was discovered and extracted from the Penicillium notatum. Now, he is Alexander Fleming. He was working on this particular one. And it is known that after the discovery of penicillin from the Penicillium notatum, the full potential of the penicillin for established, that is established by the efforts of Ernst Chain and Howard Florey. Ernst Chain and Howard Florey were the scientists who established this full effectiveness. So this is Ernst Chain and Howard Florey. Now, it is also known as that this antibiotic, that is penicillin, was extensively used to treat American soldiers wounded during the World War II. So, due to its great particular effects and great beneficial effects to the human, Fleming, Chain, and Florey were awarded the Nobel Prize in 1945 for this discovery. In 1945, they shared this particular and the particular, we can say, Nobel Prize. Okay. So this is the particularly scientist, and main work was by Alexander Fleming. Now after penicillin, it was known that several other antibiotics were also purified from the other microbes. Several antibiotics have been discovered, and in this, the streptomycin is there, gentamicin is there, neomycin is there. Such antibiotics are there. Now, if we talk about antibiotic, then we have to note that from where these particular antibiotics are obtained. So it is known that bulk of antibiotics are obtained from three group of organisms, that are bacteria, then actinomycetes, and fungi. Now if we talk about antibiotic and source, and the source from which this antibiotic is obtained, if we talk about from bacteria, the antibiotic which is obtained, its name is bacitracin, and the source of bacitracin is Bacillus licheniformis. Okay. Apart from this, if we talk about from actinomycetes. Actinomycetes is which antibiotic are obtained. Then in that, the first we will remember, that is chlorotetracycline. Chlorotetracycline, and this chlorotetracycline is also known as aureomycin or tetracycline, and this tetracycline is obtained from Streptomyces aureofaciens. Okay, from this antibiotic is obtained. And second antibiotic in case of the which is obtained from the actinomycetes, that is chloramphenicol or chloromycetin. And the bacteria actinomycetes which is involved, that is Streptomyces venezuelae. Okay, so that is what chloramphenicol or chloromycetin. The third antibiotic which is obtained from the actinomycetes include erythromycin. The source of erythromycin is Streptomyces, erythraeus. From this erythromycin is obtained. Then fourth antibiotic which is obtained from the actinomycetes is streptomycin. And this streptomycin is obtained from Streptomyces gracius. Then fifth is oxytetracycline, or we can say terramycin. And oxytetracycline or terramycin is obtained from Streptococcus ramosus. Then sixth is neomycin, which is also obtained from actinomycetes, and neomycin is obtained from Streptomyces freddi. Okay, so this all tetracycline, chloramphenicol, erythromycin, streptomycin, terramycin, and neomycin are obtained from the actinomycetes, which is a filamentous bacteria. And third, from fungi if we talk, then from fungi, the antibiotic which are obtained, that is penicillin. And we know that the source of penicillin is Penicillium chrysogenum, because nowadays commercially the maximum penicillin is obtained from the chrysogenum, and commercial application is from the Penicillium chrysogenum. The second is glyceofulvin, and glyceofulvin is obtained from Penicillium glyceofulvum. And the third antibiotic, that is fumaglin, and fumaglin is obtained from Aspergillus fumigatus. Okay, so this is the three antibiotics which are obtained from the fungi. Okay, so that is what about what we can say the antibiotic which are obtained from this different organism. Now, apart from this, antibiotic have greatly improved to treat several diseases and such deadly diseases like we can say plague, that the whooping cough, which is also known as kali khasi, diphtheria, which is also known as galgotu, and leprosy, that is kushto. Such diseases, deadly diseases, which used to kill millions over the globe have been treated by the use of this particular antibiotic. Okay, so these are the different disorders. Now today we cannot imagine the world without antibiotic. So in every disease we need antibiotic to treat them particular properly. So after alcoholic beverages and the antibiotics, let's talk about some of the chemicals, enzymes and other bioactive molecules which are involved. So now microbes are also used for the commercial and industrial production of certain chemicals like organic acids, we can say, then alcohol and the enzymes. Okay, these all are produced, we can say, commercially as well as for the industrial application by certain microbes. And if we talk about these chemicals, they are we call a bioactive molecules. Then the bioactive molecules, if we talk, what how they can be defined. So bioactive molecules are those molecules which are functional in living system or can interact with their components. Functional in living system and interact with their components that are known as bioactive compounds. And here also we are going to talk about certain chemicals or bioactive molecules like organic acid, alcohol, enzymes like cyclosporin A and statins. All this we are going to discuss. First, we are going to talk about the organic acids. And organic acids are the certain microbes, they have ability to convert carbohydrate into organic acid. So we are going to talk about the non-organism type of microbe and the particularly product. So if we first talk about the Aspergillus niger, which is a fungus, it is a type of fungus and it is going to produce a citric acid. And if we talk, the citric acid is employed in dyeing, ink, medicine, flavoring, and preservation of food. In such things, this citric acid is getting used. Apart from this, other organism is Acetobacter aceti, that is a bacteria. Now, this bacterium is going to produce acetic acid, and acetic acid is used in preparation of the vinegar. Apart from this, next bacterium is a Clostridium botulicum, which is a bacteria and that is going to produce a, we can say, butyric acid, and this butyric acid is used for making a rancid butter. Okay, then apart from this, a lactobacillus as well, we know that this is a bacterium, it is used to produce a lactic acid which is produced by the curd making. Okay, so these are the particularly organic acids. Okay, so here if we want to define what are organic acids, then this certain microbe convert carbohydrate into organic acid. Okay, there they are particularly able to produce. Now, apart from this, next we are going to talk about a yeast, and we know that the yeast which is used, that is a Saccharomyces cerevisiae, and this Saccharomyces cerevisiae is used in commercial production of the ethanol, and we call it as a beer or baker yeast. Now, microbes are also used for the production of, we can say, enzymes. And if we talk about this enzymes, then next we are going to talk about the lipase enzyme. Okay, we remember if we talk about enzymes are hardly 1.1 to 1.5% of the total enzymes are employed in industry and medicine. Out of total enzymes, only 1 to 1.5 are going to be used in the particularly industry and medicines. So now first we are going to talk about the enzymes that are known as lipases. Okay, so if we talk about the lipases, these are used in detergent formulation. In detergent, whatever detergent we use for the cloth washing or in laundry, that have this lipase enzyme, and this lipase helpful in removing oily stains in the laundry. And because of this, in detergent are able to remove these oily stains from the laundry. Now it is known that these are the particularly, we can say, the some detergents. Now it is known that lipases are obtained from Candida lipolytica and Geotrichum Geotrichum candidum. Okay, so in this, the trichosis are obtained from this particular two plants, two particularly we can say fungus. Now, bottle fruit juices, if we talk, they are clarified. If we prepare a fruit juice bottle at home, if you will prepare, we will have some fiber like structure within that. But when we get these fruit juices, bottle fruit juices from the market, they are clear and they do not have any of this such fibers. So this bottle juice fruits are clarified in such a way by the use of pectinases and proteases. So these are the bottle juice which are particularly produced by the pectinases and proteases, and it is known that these pectinases and proteases help in clarifying fruit juices and making them clearer as compared to those made at home. And it is known that these pectinases are produced by Aspergillus niger, then Bysoclemys fulvo. Okay, so from this, these pectinases are obtained. Apart from these, proteases which break a protein, and pectinases actually break the pectin. Proteases are obtained from from Mortierella renisporum and Aspergillus and Bacillus species. Okay, from this proteases are used and they clear the fruit juices. The next enzymes are the streptokinase. And streptokinase, it is known as it is produced by the bacterium Streptococcus, and it is modified by the genetic engineering. And this streptokinase is used as a clot buster. It is used as a clot buster. And it is used as a clot buster for removing the clot from the blood vessels of the patient who have undergone a myocardial infarction. Okay, because myocardial infarction lead to the heart attack. So removal of this clot buster is carried out by the streptokinase enzyme. Okay, now after this, another bioactive compounds molecules that is we call a cyclosporin A, and it is known that this cyclosporin A is used as an immunosuppressive agent in organ transplant patients. So when organ transplant is occurring, the immune system of the particular person who is receiving the organ, its immune system getting suppressed by the use of this cyclosporin A. So the transplant becomes successful, and this particular cyclosporin A is obtained or produced by the fungus that is Trichoderma polysporum. Important question that is getting asked. Okay. Apart from this, the another compounds bioactive molecules are the statins, and it is known that statins produced by the yeast that is a Monoscus purpurus, and it is commercialized as a blood cholesterol lowering agent because it's lower blood cholesterol. Now it is known that statin act as a competitive inhibitor for the enzymes responsible for the synthesis of cholesterol, and it is known that they inhibit the enzyme that is HMG-CoA reductase. So now which enzyme is that? If you want to remember that, then we remember that statins resemble mevalonate and is competitive inhibitor of beta hydroxy beta methyl glutarate coenzyme A reductase HMG CoA reductase. Okay, so this is the particularly we can say the statins are the bioactive compound which is used such a way. If we talk about some more things, we remember that some more important points that Waxman and Albert discovered streptomycin antibiotic, and it is known that term antibiotic was given by Waxman. It is known that lactic acid was first organic acid. It is first organic acid to be produced by industrial fermentation. Okay, so such a way this particular lactic acid is produced. So this is all about the what particularly industrial products like alcoholic beverages, antibiotics, and the bioactive molecules. Now, the large vessels used for the production of microbes at industrial scale that we call a fermenter, obviously to carry out the fermentation process. Brewer yeast, if we talk, it is nothing but a Saccharomyces cerevisiae, which is also called as brewer yeast and breaker yeast. Who discovered penicillin? So obviously we know that Alexander Fleming discovered the penicillin from the Penicillium notatum. Okay, so that is all about what use of microbes in the industrial products. Okay, I hope you understood well. Thank you.